My name's Tom, VP OMNiLEARN. Come on in, I'll show you some cool science labs. So we're just going to give you a little overview, a little taste of some of the stuff we can work on. That way, when we come out there, oh hey Johnny, when we come out there, we'll leave it open-ended so you can tell us what direction you want to take it in. You know, everyone's worried about health today, and with good reason. You have to watch out for high-fat foods. But what I hate is when you go to the supermarket, buy some low-fat ice cream and it doesn't taste very good. Big problem, right? Well, big solution. Scientists, through gene modification, can enhance, they can turn on certain genes. Take these glowfish, for example. They've been genetically modified to produce this fluorescent red or yellow or green effect. Simple procedure, right? I'll teach you about it. The ice cream manufacturers and their scientists have come up with a way to defeat freezer burn, even in low-fat ice cream. What they do is they went up to the Arctic Circle and found the ocean poot fish. The ocean poot fish has a gene, it's an anti-freezing gene that makes a protein so that they don't freeze in the cold Arctic waters. What they did is extract that gene, produce the protein, and take the protein and put it into the low-fat ice cream. So instead of making these gigantic large ice crystals that cause a freezer burn on rough surfaces, now they have these very small, smooth crystals that give it a creamier, richer taste. What does all that mean? Well, the end result is smooth, delicious, low-fat ice cream.
Hey guys, Tom's on here right now, but it gotta be quick. Now you see these fish? They've been genetically enhanced to brighten their colors. Now quick, I want to show you something. Now I'm going to give you a quick look how to genetically modify a fish egg. Okay, now I want to show you one more thing.
Hey guys, now this is a microcentrifuge. A microcentrifuge is really good at separating liquids by density. Let's say you have a sample of saliva at a crime scene. I want to separate the cells from the saliva. And eventually, I want to separate the DNA from the cheek cells. This is basically a fancy Xerox machine. It's called a thermal cycler, and instead of making copies of documents, it makes copies of DNA. You are a forensic scientist working on a murder case. You get a sample of DNA. Good idea would be to make some copies and send those copies all across the country, all across the world if you wanted to, so you have a lot of different eyeballs looking at the same piece of evidence, and if you make a mistake, it's not the end of the world. We want to bring the equipment out to you, and that way, one day when you're working in a research facility and your manager shows you a thermal cycler, you can say, hey, I know what I'm doing. I've done this before. You have the knowledge base. You have the comfort level. When you're sitting in the jury box and you're trying to decide whether someone's guilty or not guilty, you'll know what DNA fingerprinting is. You'll be able to make a judgment on a crime, no matter how small that crime is. It could be something as trivial as someone took something from you. Something you were waiting all day for, looking forward to it. Looking forward to some smooth, delicious, low-fat ice cream.
So Nick, what is DNA fingerprinting?
DNA fingerprinting is the most current high-tech way to solve crimes. This is the DNA found at the crime scene. Okay, we have a spoon, and we've taken the DNA off the spoon and loaded it into one of the tubes. Next, I've taken DNA from the suspect. Okay, we're dealing with Johnny. Johnny's DNA is in the tube right now, and we're going to try to match it up with the evidence found at the crime scene. Let's use this technology to help Tom out. Each well is going to have its own specific pattern because we're using different DNA samples. Now we have some electricity running through here, and what that's going to do is push the DNA with the dye in each well through the gel. This box shines UV light through the gel to better see the DNA patterns. This is a picture of the gel.
Now we've been throwing a lot of things at you, and very quickly. Sometimes it's better to just slow it down, get back to the source. Let's meet the OMNiLEARN founder, Jane Powell, and see what she's up to. What are you working on?
Well, I'm working on trying to solve the big problems. Alternative energies, starvation, HIV.
Is there any hope?
There's so much hope. Teaching cells to self-replicate and become little tiny factories in your body. They're custom designing medicines. They have oil-eating bacteria that actually know how to break down the hydrocarbons into smaller molecules. The creatures digest them and then actually excrete new fuels for us, methane.
How good is that?
And the common theme between everything that I've been researching seems to be proteomics, three-dimensional, lock and key, all about shape.
Good stuff, Jane. Always fun to see what you're researching in the library. Now, let's get back to the lab and see the results of that crime gel.
Both patterns have come out. Now, can you match up some patterns on here?
Okay, well I see number 3 is the evidence.
Right.
And the one that looks exactly the same would be in well number 6, which was
Johnny.
A little demonstration about how vibrations in the air can cause serious damage. Just wait till Johnny gets here. Hooah! Alright, we appreciate you guys hanging out. Hope you enjoyed yourself. Hope you got a good taste of some of the things that we're into. Most importantly, we're looking forward to seeing what you're into. See you soon. Bye-bye. This is your basic hydrogen fuel cell car. You have these protons, these hydrogen protons.
To the river, I take my old rocking chair. If the blues don't take me, I'm gonna rock away from here. Oh, troubled mind.